So, good morning and welcome to my podcast, Pushing Boundaries, about pioneering research, breakthrough discoveries, and unconventional ideas. I'm your host, Dr. Thomas R. Verney. My guest today is Nirosha Murugan, PhD, assistant professor at Algoma University in Ontario. And as she told me, it's in Northern Ontario, close to Sault Ste. Marie. Have I got that right?
All right.
Okay. Dr. Murugan is an interdisciplinary scientist with a strong passion for research and teaching. She studies regenerative medicine and cancer biology. She investigates how cancer cells can be reprogrammed into healthy cells using regenerative microenvironments, and in turn, how cancer microenvironments can modulate the function of distant organ systems, such as the brain, to induce cancer-related cognitive and neurological impairments. Welcome, Dr. Murugan. Welcome to the program.
Thank you so much for having me.
It's a total pleasure. The paper, that first drew my attention to your work was your 2020 paper, Mechanosensation Mediates Long-Range Spatial Decision-Making in Aneural Organisms. And you co-wrote that with Dr. Michael Levin of Tufts University. Am I right?
Yes, I did.
And so when was it that you were at Tufts University? How long ago was that?
So I started my postdoctoral fellowship with Dr. Levin in 2017, and I just finished with him in 2020, but we are still collaborators, and he will always be my mentor.
Well, he's one of my heroes, absolutely. I'm in total awe of this man's genius, really, and we are going to
definitely a pioneer thinker for sure.
Amazing. I was first attracted to his work on planarians. Did you do any work on that yourself?
Yes, I have. A lot of my work that I explored with biophysics was in planaria in my PhD. And a lot of the expertise and the ideas that I've had in my graduate work is what attracted me to Dr. Levin as well. Which got me recruited into his lab because we both had the same way of thinking and we had similar ideas to approach complex problems.
Wonderful. So what is it that you're working on now?
So right now I have a couple different projects, but they all fall under the umbrella of trying to control biological systems from a top-down approach through biophysics. So my understanding of how biological systems work is that there needs to be a unified language that allows systems to be controlled effectively. So one of the projects that I have focuses more on the neuroscience aspects and controlling brain activity, and the other project focuses more on controlling cancer and regenerative systems. So I'll go through the couple projects if you don't mind.
Yeah, okay. So let's talk about the first one first. You did your research on the Physarum polycephalum, which is simply a slime mold, correct? Right. And so what is it that attracted you to the slime mold?
Well, actually, I didn't know anything about slime mold when I started with Dr. Levin in 2017. We both had the idea of, again, trying to control biological systems using biophysics. It was either bioelectricity, biomechanics, biomagnetics, electromagnetics. So we wanted to use a system that was, high throughput that we can reproduce in the lab and have control over. Mm-hmm. So we explored the idea of cognition. What is cognition really? And if we had a system that had cognitive principles but didn't have the brain, can we use that system to explore what minimal cognition is? And that's kind of like where we entered the world of Physarum.
So it's really thinking without a brain that you're looking into.
That's exactly it. Because we came up across a paper where they showed minimal cognition in snails. But snails, as you know, still have a nervous system. So we wanted to take that a step further. Let's take the brain, let's take the nervous system out of the equation and see what we can get in terms of a cognitive organism.
Hmm. And so what did you find out about the slime mold? What sort of things have you found out?
Sure. So we know through literature that they are able to solve complex problems like trail navigation, creating shortest networks, things like that. I want to kind of take this in line with conventional ideas of what cognition is, which is decision making. So we wanted to create a assay where we can assess decision making very simply. We give two tasks or one task where they have two options, and the Physarum needs to pick one of the two options.
Sorry,
Traditionally, in mammalian cognition, traditionally in mammalian research with cognition, it's usually through some sort of food source or drug source. And how do you assess food and sensory in an organism that doesn't have a traditional sensory system? So that's where biophysics came in, and we used the idea of mass instead of a chemical stimuli. So we had two masses, one of low mass, high mass, and we had the Physarum essentially decide which mass to pick.
Why would it be attracted to an inert mass? In other words, not food. Why would it go? I think there were a couple of discs that you used in your experiments, correct? So why would it be attracted at all to a mass, to an inert subject?
Yes. If you're kind of looking at a sensory stimuli like food, all organisms, regardless of whether it has a brain or not, is attracted to some sort of nutrient. Right? And with nutrients are usually coupled to mass. The more food there is, the more mass there is in the environment. So let's take one of the factors out, which is the chemical stimuli, and let's just use the mass as the sensory stimulus. And so that's where we pick the mass.
Can you tell us a little bit about that experiment that you describe in the paper that I just mentioned?
Absolutely. So how the experimental paradigm works is we have a 10-centimeter petri dish with a small, consistent amount of Physarum slime mold placed in the center of the dish. And equidistant on either side of the Physarum, we have a mass, either one disc, which is considered to be low mass, or three discs, considered to be high mass. And there's no chemical stimulation or chemical stimuli in its environment. And the environment itself is an agar dish, agar plate. And we allow the Physarum to explore and pick a side within a 24-hour period. And we image every 15 minutes over that period until it kind of create like a time lapse of what the decision is of that Physarum.
I understand that it moves very slowly, like 1 millimeter an hour.
Extremely slowly. So behavioral experiments on a Physarum is tests your perseverance and your patience for sure.
Yes. So what did you find out?
So we found this was a very interesting finding that we found was it takes about 4 hours for the Physarum to quote unquote decide. And what I mean by that is when you actually look at the video, the Physarum for the first 4 hours actually grows equally in both sides, in all directions, 360. And then after the 4 hours, does it have a unilateral branch towards the heavier mass? And we've experimentally shown that across hundreds of plates, and showed that about 80% of the time, the Physarum usually picks the heavier mass. And that's what we showed initially. And we've done in that paper, we've shown chemically and physically and through computational modeling that this is through some biophysics information processing of the Physarum in its environment.
You say 80% of the time. So are these different slime molds that you're using, or is it the same slime mold over and over again?
These are different funds. So we have done, taking a master plate or the big plate and kind of reculturing the same strain of slime mold, but we have shown that we can take newer slime molds or of different ages and or different sources and still see the same phenomena.
So do you call that thinking? Would you by that as thinking?
We define thinking as, sensory information coming in, processing with some kind of a motor output, which is what conventional, cognitive scientists may call thinking. I think so. There's a period of time where the Physarum is processing the information of its environment, and it's a reliable time. So I think in that period of time, it is thinking about what the next movement or what decision it needs to make. So from that rudimentary definition, I think it is thinking.
Right. So Dr. Levin, Mike Levin, that you worked with, I have a quote here from him. I'll just read it to you. Our discovery of the slime mold's use of biomechanics to probe and react to its surrounding environment underscores how early this ability evolved in living organisms and how closely related intelligence, behavior, and morphogenesis are. I guess you would agree with that?
Absolutely. Absolutely. It just shows how evolutionary consistent thinking and decision making are. What a Physarum does in terms of just, it's very simple, it's very rudimentary in terms of what that decision making process is. The mechanism might be more complicated, might be different, but I think the actual process is the same.
Okay, well, that's great. So in terms of yourself, like, was there anything in your own background that kind of got you sort of on this path and towards biology and exploration of what I would call cellular intelligence? But you probably call it something else. Is there
cell-based intelligence or collective intelligence is more of what I'm interested in. Collective intelligence, and this is where that led me to kind of be connected with Dr. Levin in my PhD, is cancer is a problem that has been existed for many decades, many centuries. And I think the way that we look at cancer, we can kind of gain some insight of how we look from collective intelligence. I feel like cancer is an intelligent organism. It's survived, it has made decisions, whether they're higher level or not, to survive in environments, biological environments that may not be conducive for them. So cellular intelligence from a cancer perspective is a very interesting topic, and that's kind of what led me to Dr. Levin and kind of exploring this from a Physarum perspective.
When did you, at what age, how far back did you become interested in cancer?
Actually, my master's. So I don't know how old I was. Early 20s, I would say. Early 20s. I was always interested in biophysics as a control mechanism for language. And I think the collective intelligence of how we look at communication intelligence, in my opinion, go hand in hand. How well you're able to communicate with your environment and your neighbors is important for intelligence. And we typically look at communication through a chemical perspective. That's how cells talk to each other, organisms talk to each other using molecules. But I think a missing piece, a big piece of that communication is biophysics. It could be bioelectricity through chemical use, like Dr. Levin has shown with ion channels and gap junctions. Another area that I'm exploring is more on the quantum side, quantum biology side, through electromagnetics and photonics as well, and see how all of those pieces come together for collective intelligence, in cognition. And when collective intelligence goes awry, you get cancer. And that's kind of exactly what I'm doing right now in my lab.
That's a wonderful explanation. In terms of your own development, was there anything in your childhood, in your early teenagehood or childhood, that kind of already sort of pointed you in this direction?
Well, cancer personally, in my family has unfortunately played a devastating role. We've lost several family members to cancer, and I, also have been afflicted by this disease as well. And it's just looking into it from a biological perspective, from a scientist perspective. It's I feel like we need a different perspective on how we talk about cancer, not even just describe it. People describe it as a devastating disease and infiltration, but again, if we look at it as an organism of its own, with its own intelligence, with its own goals, then we can kind of think about therapies around that. So very early on in my life has this been a question, and I've always been fascinated about, why is this such a problem that no one's been able to solve? That's the thing that started on very early in my life. And that drove me to where I am today.
Okay. So what was your favorite subject in school?
Oh, interesting. Oddly enough, calculus.
Really? Oh,
really?
keep me away from calculus.
Yeah, in high school, I was the only one who willingly took all the different calculus courses. And then even in early university, I willingly took calculus.
That's amazing. That's amazing.
Yeah. And also statistics, which is kind of makes sense with the interest in math. I love stats, absolutely love statistics. And having that, I think I was programmed to kind of design experiments and design things that make me, I think a decent scientist.
So along this path, which so far sounds like very successful, have you come across sort of any difficulties when you were pursuing this subject? Any problems? Any I don't know, obstacles that you had to overcome?
Barriers?
Yes, barriers.
Yeah, actually, absolutely. Funding barriers, barriers to acceptance of an idea. It has been a very, very big one until recently. And I think Mike has done a really good job at taking frontier ideas and presenting them in such a way that, everyone can understand and run with. And I've learned that in my postdoc with him is that, frontier ideas will always exist, but designing an appropriate experiments to prove them or disprove them and presenting them in the appropriate way is extremely important to progress.
Yes.
And that's a big barrier that I faced for a long time, and I still face. And again, it's going to take a lot of work to prove and disprove appropriately and present appropriately as well.
So do you feel supported by the University of Algoma, where you are at the moment?
Yeah, I do. I've been fortunate enough to be funded very well in the area that I am exploring. To the point, also, like I have been nominated and successful in becoming Canada's research chair in biophysics, tissue biophysics. And I think that tells me that, our scientific community and our clinical community are open to frontier ideas. And along in that vein, I've just received the New Frontiers Research Fund, which funds high risk, high reward projects.
Wonderful.
And being able to be funded by that pool and by that community gives me confidence that these barriers can be overcome with some perseverance and some support with the community.
Yes, you need support. Speaking of support, are your parents still alive?
Yeah.
Do they understand and support what you're doing?
They do. It took a while. Culturally, I was encouraged to get into the medical field, and also, like my grandfather was a practicing physician in my back home, and that's kind of where the spark of understanding and questioning physiology probably was instilled into me from very young age. And, it was always expected that I would follow in that path. But I was always interested in challenging and developing applications that could help at a global scale rather than at the individual scale, which I feel clinical practice is more on a one-to-one treatment level, whereas clinical science has the potential to be at a more large scale effect. So it took them a while, but they see it now. They see the importance of science and they see the importance of research and the impact that it can have to not just the clinical world, but to also how people think about a certain topic.
Mm-hmm. Very, very interesting. Are you in an intimate relationship at the moment?
I am. My husband actually is also a professor here in the Department of Psychology. He's a neuroscientist.
Oh,
He was.
what
Sorry?
what does he do? Your husband? What?
You will be very excited about his his research program because he was co-supervised by Dr. Levin at Tufts and primarily supervised by Dr. David Kaplan in the biomedical engineering department. So he is extremely interested in minimal cognition, and he just received an NSERC, a federal grant, to develop brain organoids to assess minimal cognition. So connecting brain organoids to robots, giving them a motor output to assess what the fundamental limits of minimal cognition or cognition are.
Amazing. Well, you have quite a family. Do you have any children?
I do. I just have a toddler right now. She's 2 years old. And she was born at the end of my postdoc as I transitioned to a new PI. So it's been a new professorship, new motherhood. Everything has been a learning opportunity. And also going through this during the pandemic was also a new learning opportunity.
Yes. Would you like your child or children to follow in your footsteps?
I've had this question in the back of my mind since I was pregnant. And the answer is yes, because I think this career path gives you characteristics and traits that are helpful lifelong. It allows you to question how progress is being made in society and question yourself and, give you skills to tackle those questions as well.
Was it difficult to settle in Sault Ste. Marie or wherever it is that you are?
Yeah, it has been difficult. It has. Again, primarily because of the pandemic. Moving in the middle of the pandemic from a very large city like Boston to a very small city like Sault Ste. Marie, and to give you an idea, it's only about 80,000 population size. So navigating that move and kind of understanding the landscape has been difficult, but now it's very exciting. Sault Ste. Marie is in a community, is a city more environmental based, so a lot of outdoor activities, much different than Boston.
Okay. So how does this work, do you think, or hope, applies to cancer and cancer therapy?
Yeah, so kind of looking back at what I was saying about the collective intelligence. So we all know that cancer is a devastating disease. It's a healthy tissue, healthy cell that has essentially gone haywire. And it has its own goals and directives of how it's going to survive in a microenvironment. And how I look at this is if we can tap into what the cancer cells are saying differently from the healthy cells, then we should be able to talk to them directly and change their fate back to the healthy cell that they once were. So that it's very anthropomorphizing the cancer itself, but that's the idea.
That's wonderful. That's really interesting. Interesting. Yes. So I know Michael Levin is probably one of the people who has influenced you the most, but tell me about two other people in your life who you feel have really had a strong influence on your life.
Absolutely. Before I even met Dr. Levin, my graduate supervisor Dr. Michael Persinger, has been an enormous inspiration to me, enormous inspiration. Outside of the science world, just academically, he provided the freedom as a graduate student to think and ask questions, and provided me the skills to ask and test those questions appropriately. And I'm so grateful for that opportunity. He was also, so he's a neuroscientist with unconventional ideas and approaches to how he solved problems. And so he's an electromagnetic biologist and neuroscientist by training. So that's kind of, he's my number one inspiration in academia. And he was the one who led me to Dr. Levin.
Right, and one more?
One more has to be my grandfather. Again, him being a physician, he was a physician, and later on in his life, he kind of tied cultural principles into science. Tested the idea of what is religion from a brain activity point of view. You know, I'm not sure if you've heard of the conversations around the God gene. You know, is there a God gene? Is religion a natural selection mechanism? Does it follow natural selection? So those kinds of like high level questioning, and having a South Indian background, you know, talking about what consciousness is, consciousness with brain activity, kind of at a really young age, helped me appreciate, you know, the limits of the brain and the body.
Mm-hmm. What do you think consciousness is?
Oh, that's a deep question.
Let's look it up.
So I should also mention my husband. He's extremely interested in consciousness, and he recently wrote a paper and won an international grant to scientifically provide evidence for consciousness existing outside of the brain. So we've had many conversations about what we think consciousness is.
Right.
Consciousness, in my opinion, is a regulated electromagnetic state.
Uh-huh.
That's how I would describe what consciousness is. And our brain is a conduit for processing this electromagnetic information. So we know radios can do the same thing. Mm-hmm. We have magnetic waves across the universe. Mm-hmm. And I think all the neural activity that we have from the action potential produces electromagnetic activity that has information within it. So if I had to kind of distill that, consciousness is just a regulated source of information through electromagnetic. Is just a conduit to understand and experience that information.
Mm-hmm. Mm-hmm.
Yeah.
Yeah, well, that's a mouthful. Yeah, I'm thinking about that. And what about the mind? How do you sort of think of the mind?
The mind and being conscious and consciousness are almost entangled. You can't really separate them. The mind is where you have more of the hardware. And if we think of consciousness as the software, mm-hmm. It's something that it's hard for me to describe. I'm more of like the hard biologist, but I think the mind is, again, that intimate connection between this information that exists as consciousness, which, you know, some people can say that can exist outside of biological tissue. And the mind is it being brought in to biological tissue to process and perceive.
Mm-hmm. Mm-hmm. Okay. Well, you know, it's always an interesting question, and every person has a different answer to it, right? Yes.
Yeah, I guess your own experiences and your background will help you answer that.
Right. So, in your life, you're what, about 30 years old, approximately?
I was 33.
33, okay. So in your 33 years, do you remember one time when you were truly, absolutely happy?
a lot of information to
Yes.
process. Um, yes, actually, I can.
Tell me.
It was oddly enough a day during the pandemic when
Yeah.
everyone was stressed and worried about their family. Uh, I was happy that I had a healthy daughter and I got to watch her grow. If being in this career state that I am and starting a new job as a scientist and a researcher at an institution in Canada, it's very demanding.
Yes.
And being at this stage in my life, being a Canada Research Chair, having a healthy child, just being grateful for all of that. And honestly, being able to be a Canada Research Chair in an area that I'm extremely excited about and can make a difference.
Mhm.
I think all of that ties down to being grateful was the fundamental point where I can say that I was the happiest.
And when was that? I mean, was there one moment? Was it when you received your nomination or your award?
I would say it was much after that. I mean, I of course that day I was very happy, but I think sitting in the house that I was in my home and reflecting on everything that's happened. Oddly enough, that day came after the loss of my grandfather.
Oh,
So it took a loss for me to be happy, to understand that happiness, because he did live a very good life. He lived a great life, and to reflect on the life that I have because of him, essentially, um, made me extremely happy.
hmm. I noticed you have a picture of a heart behind you. Is that a human heart?
It is a human heart. It's a painting done by my husband, Dr. Rouleau.
So he is not only a scientist, but also an artist.
He is an artist. All of that art, actually, you'll see that
Yes,
there's some mural paintings back there. I don't know if you could see it.
yes, I can. Yes, yes.
Yeah, that's the and that's digital art of a brain section of an actual human brain section that we work with in our PhD.
You must be the only person in the world who has got a painting of a brain behind her.
Yeah, it's a good reminder
Yeah, of course.
of the intimate tanglement between love and science.
Yes, yes. How do you relax? What do you do for relaxing? Do you do any art?
And I play music. I'm a Carnatic musician, so I play. It's a South Indian instrument. It's similar to the sitar, but it's not. It's called a veena.
Oh, do you have it handy? Can I see it?
I'm actually in the lab right now. I don't have it with me. It's at home.
Okay. So
So
how long
music relaxes me
how long have you been playing that?
since I was 7.
Oh, so
Very long time.
you must be pretty good at it.
Okay. My so Nicholas is also a drummer. So he's a really good drummer. And he's more on the Western music side, and I'm more on the Eastern music side. So we have some fun playing music together. We also we're very competitive, so we like gaming.
Uh-huh. You like gaming.
Yeah, tradition like not particularly video games, but more like board games, things like that.
And you said we are very competitive. Does that mean you're competing against each other when you play games?
Absolutely. So there's no other way to play games.
Okay, all right, all right. So, um, what is the best advice that you ever received in your life?
I know exactly what it is
What?
because I give the same advice. It was another mentor of mine, Dr. Seema Kumar, who's uh, immunologist, more works on what sepsis from my graduate school. He told me take every opportunity don't look for them, but just take it. Like, unrelentingly go for it and take it. And I absolutely have been doing that ever since I heard that from him. And it's not just in my career, it's in my personal life, as well. And also just within myself as well. So opportunities to better my own thinking, better my own health. Those are all opportunities that I have to take the initiative to go get. So that's the best advice.
That is good advice. So if you won $10 million tomorrow, what would you do with it?
I would build an institute, probably not enough money, but I would build an institute and bring all of the frontier people together to do more cool stuff together.
Wonderful, wonderful. So what's in the future for you? Where do you think you are going from here? Are you going to stay at Algoma for a while, or are you going to move on?
Well, that's the plan. I'd like to, Algoma, the reason why I came here from a big institution was that I grew up in a very big city. I grew up in Toronto. And I did get into U of T, into the neuroscience program, but I chose to go to Laurentian in Northern Ontario because I wanted that one-on-one ability with my professor, wanted to get that connection with my peers. And I would not have gotten the access to research. In my PhD, I helped lead a $1 million grant by myself. Right. Which most people don't get. Right. And I came here to Algoma to bring all the expertise I have to another person who would have been like me. Yes. Who's just as brilliant, just needs the opportunity. So I'd like to stay here for a little bit longer to be able to do that. And if I have to be elsewhere to do that, and that's where I go. Where did you meet your husband? In graduate school. Where? In graduate school, in our histology lab, cutting tissue.
Oh, I see. Very romantic.
Yes.
Okay, so before I let you go, I'm wondering, I know you're only 33 years old and it's kind of early to think of that, but what would you like to be most remembered for?
Absolutely know this question. Know this answer is I like to be known for the person who did the thing that no one else said that they could do.
Which is what?
So, taking biophysical language and changing biology. So taking biophysics, taking physics, and changing biology. That's exactly what I'd like to be remembered for.
Wonderful, wonderful. Well, I hope you will, and I hope you will you will live many, many, many years to achieve that. And I wish you much success.
Thanks so much for this opportunity. It'd be great you've asked some really stimulating questions that I thought about before, but really talking it out actually helped navigate what I should do next.
And clarify, perhaps.
Absolutely.
Well, thank you for joining us. May I call you Nirosha?
Absolutely.
Thank you. This was totally fascinating. I just want to say my guest next week will be Swiss psychologist Klaus Capelli, with whom I will be discussing his psychotherapy experiences treating children born by in vitro fertilization and other third-party conception techniques. So that's next week. And today, once again, we had Dr. Nirosha Murugana with us, a professor at Algoma University, making huge strides forward towards perhaps ending this terrible cancer epidemic that we have been suffering of for many years. Thank you, Dr. Murugan. Bye-bye.
Thanks so much for the opportunity.